Hello everyone. We're really happy to have you guys here for another PRC session. Today we're going to have a special talk where we're featuring Mr. Shayanatan Banerjee. He recently completed his master's in computational biology from IIT Madras. He was always fascinated by how one can use computational modeling of genomic data to understand the underlying biology of human diseases. And he's also a huge fan of community-driven open source bioinformatics software and participated in several hackathons and open source summer projects like the Google Summer of Code 2019. Today he will talk to us about his work on an AI algorithm that can detect cancer mutations, which was recently published in the International Journal Cancers. Apart from his research, he will also give us an idea about how AI and ML can be applied to biological data sets. We're all really excited to listen to your talk, Shantan. Everyone, please switch off your cameras and mics during the talk. If you have any doubts, please feel free to unmute yourself and ask it out or type out the questions in the chat box. Over to you, Shantan.
Thanks for the introduction, and thank you to the Biotech Research Club for hosting me and letting me the opportunity to present my work. So this was my master's project, and this recently came out, and we were quite excited about getting this publication and specifically this tool that can detect cancer's mutations using AI techniques. So before I start giving the talk, I just want to let you all know that please feel free to ask questions at any point of time, and I'll start with a generic overview of AI and then move slowly to the technical aspects of my work, and then again move to a generic introduction to all the AI-based work that's going on in our lab. So yeah, the talk is titled "The Devil is in the DNA." So we were specifically interested in finding patterns of mutations or patterns of DNA sequences that surrounds particular mutations in the human genome from cancer patients. And I divided my talk into three parts. So these are the three major themes. So the first part is machine learning and AI in healthcare. So this would be a very broad, generic introduction to the field. This will be followed by the tool that we recently developed called NBDriver or neighbourhood driver. So it's a cancer mutation prediction tool using AI techniques, leveraging natural language processing techniques as well. And finally, I will end by giving a broad overview again on precision cancer genomics to sort of motivate you to pursue further research in this area, with a specific focus on all the exciting research that's going on in our lab. So I would assume that people are moderately familiar about the terms. Like they have heard the term machine learning, they've heard the term genomics. So I'll just give a very brief overview of what machine learning is. So this is by no way a fair introduction to this huge area of research. So machine learning is you have some training data. You can have training data from, it depends upon the domain that you collect the data from. So you have some training data. So you build a model on the training data. You adjust the model parameters based on some test data that you've set aside in the beginning. And you keep adjusting the model parameters to reach the final prediction model that would give you the best results in terms of a specified bunch of metrics such as accuracy, which is one of the several metrics that is used to test how your model is performing. And this is a cyclical process. You keep on doing that unless you reach the desired outcome or desired level of accuracy based on your domain knowledge or based on your research statement that you found. So machine learning specifically to healthcare, I always like to start with this example, which is this Apple Heart study, which is a recent study that was conducted from Stanford Medicine, and it involved around 400,000 participants that owned an Apple Watch, and the specific study was aimed at calculating whether a particular patient would suffer from atrial fibrillation, which is a type of which causes an enhanced heart rate. This is followed by an elevated pulse rate as well, and this some cases leads to a stroke. And what they found was something very interesting, that just by looking at the data that they collect from your watch, the underlying mechanisms which are not very clear to me. So this is a very broad study. I would encourage you to. I've left the link at the end. I'd encourage you to look at the paper. So they enrolled 400,000 participants who owned an Apple Watch, and they did this study and found that around 84% of the patients or the enrolled individuals, not patients, the enrolled individuals were found to be accurate. So this was the accuracy of the model. So 84% of the times when this particular model predicted that a person would suffer from atrial fibrillation in a specified period of time in the future, they actually had an episode of atrial fibrillation. So this was why this is significant, because because of the size or the scale of the study. It involved 400,000 participants. Of course, there were several criticisms because this was not a randomized clinical trial, which is sort of the gold standard for any medical research. But still, this is one of its kind study, and if I'm not mistaken, this data is online. So smartwatch or activity recognition is a very active area of research specifically to precision health. And if you're interested, you can go and look at this paper. So this is one of the several examples that one can give. I can spend an entire session on how machine learning can impact healthcare. But what we are really interested in is not clinical aspect of this work, so but the genomic aspect. So what do I mean by analyzing large multi-omics data sets? Again, I would assume that people know the different omics level that is very prevalent in biological researches. So you start with these different levels of omics data: is transcriptome, metagenome, epigenome, genome, etc. So all this different levels of omics layer adds information to your study. So you can integrate this data in several different ways. Although this is easier said than done. So integration is a very active area of research again, because not all patients have data for all these different levels, and not even if you have, then there are different levels of heterogeneity in each of these omics layers. So microbiome has a different level of heterogeneity and comes with its own set of challenges as compared to metabolome and genome and so on. So once, say, you have integrated this data, what you would look for is patterns or signatures or recurrences of closely related groups in the data, which you can further analyze and sort of build do therapeutic intervention among this set of patients that you have initially started with. So this is one of the very active areas of research that we are involved in, analyzing multi-omics data sets. What I would talk about the genome layer only, because that was what I concentrated on in this research. But there's some other work that's going on that has integrated transcriptome, genome as well, and sort of tried to predict whether particular genes would be cancerous or not. So this is again a broad overview of how to jump. Like, this is a clear jump from this clinical part to the genomic part, because we would be dealing with the cancer genome. So there are different genomic databases, which I'm sure most of you are familiar with: NCBI, PDV, and so on and so forth. This brings together different sources of information, and there are packages, there are abstractions that are built on top of these databases that links these databases to popularly used programming languages like R, Python, and so on. So you have these different databases, and there is an abstraction there that helps you query these databases in a user-friendly manner, so that you can download or extract specific portions based on your initial hypothesis, and you build models on top of that. So there are different. So this is by no way complete picture of what different types of databases are, but this again the different RNA types, the long non-coding RNA model organisms, and genomic resources that one usually use when dealing with this type of research. So before I move on to the next part of the talk, which would be specifically like directed towards cancer genomics, I'd like to take a moment and talk about this book by Siddhartha Mukherjee, "The Cancer, the Emperor of All Maladies," which also is also translated into a documentary film. So he won a Pulitzer Prize for this, and I would encourage you all to go and look at this book, which has dedicated an entire chapter to how computational techniques is transforming cancer genomics in particular. So this is a intimate history of what cancer genomics is. Like, the history, it starts off with Hippocrates and in the medieval age, and he would go towards, like, he would talk about all the different advances that has happened over time, and talking about the latest advances in cancer immunotherapy and so on. So again, if you're not interested in reading the book, although I would encourage you to read the book, because it has some lot of more information as compared to the documentary. But yeah, so this book has a history of how computational techniques has transformed the area of cancer genomics research. So that was my initial motivation to show this slide. So moving on, I would now move to the cancer genomics part. So this is where we'll go into the details about the technical aspect of my work. So cancer mutations. So cancer is caused by the accumulation of somatic mutations over the lifetime of an individual, right? So this can be due to different causes. This can be due to the intrinsic mutational processes. For instance, there are some DNA duplication errors, replication errors. There are environmental exposures, say exposure to specific mutagens, and this could compound over time and bring forth different sorts of mutations in the body, some of which causes cancer to progress and are known as driver mutations. So driver mutation specifically gives cancer a selective advantage to grow and progress, while passenger mutations are neutral. So this is one of the ongoing areas of research which is very active. How to distinguish between these two classes of mutations? Because just by looking at the substitution type, whether a cytosine is getting mutated to an adenine and so on and so forth, you cannot really make out whether it's a driver or a passenger. You need to have some sort of background information, a host of different factors that contribute towards a particular mutation becoming driver, or contributing towards progression, or passenger, or becoming neutral. So most mutations in cancers are passengers. So this is kind of the major issue that we face while designing our study and more specifically our experiments, because most of the mutations that are present in cancer are passengers and a very few number of mutations are drivers. So why is this a problem? Because if you are familiar with machine learning, there is this data imbalance issue that is very prevalent in biological data, biomedical domain. So if there's a very like a 10% imbalance, a 20% imbalance in data, you have to specifically design your algorithm in such a way that can handle the imbalance. Right? So your algorithm should, by principle, act on any set of variants, any set of mutations, not specifically the mutations that you have trained your algorithm on. So this is a problem that we face. So what is shown in the slide is uh, it's not that complicated. So it's the number of mutations in different cancers. So it's taken from this Lawrence et al. Study that does a comprehensive analysis of the different mutation frequency among different cancers. So you can see that some mutations, so in case of acute myeloid leukemia, childhood cancers have less number of mutations as compared to melanoma or skin cancer, where there are a lot of mutations. And so why this particular graph is important is because this helped us to choose the type of cancers that we would use in an analysis. Because again, if you're training your data set of mutations, you would definitely like to work definitely like to work like your algorithm to work on different forms of cancer and not specifically for a given cancer type. So that's what our motivation lay. So we wanted an algorithm that would be a pan cancer model that would be generalizable across different data sets, not a particular data set, and that would be robust in terms of the different metrics that I would talk in later. And this is also an extension of the previous slide. So again, a lot of mutations of passengers are neutral. So this particular study that was done, American Association of Cancer Research, that studied around 78,000 mutations from this many patients, and they concluded. So this particular graph, what they concluded was these are the different genes and the mutations. So they observed this G12D mutation in the KRAS gene, and they found that around 1724 patients had that mutation. Again, BRAF, V600D. So these are all hotspots, the regions in the genome that are hypermutated, and these specific genes are also very much prevalent in almost all forms of cancer. Again, KRAS, PIK3CA. So these are all very well-known cancer genes. So what they found was very interesting. Like these are all the mutations in these genes which are hypermutated, but they also found around 60,000 singletons or mutations that occurred in only one patient. So these are the rare driver mutations that occurred in only one patient, that was observed in only one patient. So this further complicates the problem, right? So you have a lot of passengers, and then you have drivers. Even among drivers, there are some drivers that occur very less number of times as compared to the others. And your algorithm should also be able to pinpoint those rare driver mutations with sufficient accuracy, right? So so these are the 1,000 most frequent mutations, and this graph shows that there is a lot of noise and very less signal in the data that we started off with. Was there a question? Okay, I'll move on. So again, so this is one of the main characteristics of cancer mutation data. So once you're working with this kind of data, you have to be very careful about the type of model you build, because these are the problems that comes with it. So there are different computational techniques that have been developed over the years. Like I said, this is a very active area of research, uh, and there are different tools that have been developed that can be grouped into all these different types. So there are these tools that solely depends on recurrence, or there are these tools that calculates whether a particular mutation is occurring more than what you would expect by chance, or whether they are frequent across samples, and based on that they would conclude that okay, there might be some reason that they are evolutionarily conserved across the samples, so then they might be functionally relevant, so let's label them as drivers. So these are, again, there are the structural clustering tools, functional impact, machine learning. So these are all the different tools that can be grouped into these categories, uh, and what we wanted to work, like concentrate on, was to look for a particular category, like develop an algorithm that would encompass the shortcomings of all these individual categories. So in case of recurrence, if you look at the tools that are solely recurrence based, those tools won't be able to capture the singletons, right? So because those tools are frequency based tools, those tools will look at mutations that are more frequent than random, right? So we did not want those, our algorithm, to suffer from that kind of a challenge. So uh, uh, our primary motivation came from this paper by Alexander et al. That was published in 2013, and so the signature of mutational process in human cancer. So what this paper did was they looked at mutations and one nucleotide upstream and downstream from the mutated position. So basically, you will have four possibilities here, four possibilities here. So 4 times 4 times 6. So there are 6 possibilities here. So they built this mutational catalog of 96. So basically, they would have this catalog of 96 different data points, and they would calculate what are the frequencies of each of these individual data points. So it will become clear in the following slide. So this, they fed this into a feature extraction tool called non-negative matrix factorization, which is very similar to any feature extraction tool. So there would, so if you have a lot of features, you need to condense those features into the most descriptive features, such that that has the most maximum information, that would contribute towards the maximum information for your model, right? So it's a feature extraction technique, and what they found was, so like I said, there would be 96 different points in the x-axis, and what they found was there were certain signatures that were very specific to specific forms of cancer. So why is this relevant? Is because they did a cancer type specific analysis, and they found that this signature, which is also called signature 3, has an abundance of this particular groups of mutations, or context rather. So this x-axis is basically A, and followed by the reference and followed by A. So ACA, right? So again, CCC, GC, G, and so on, right? So in the middle, they have the reference nucleotide, one nucleotide upstream, one nucleotide downstream. So that's how they calculated the catalog, and these are the mere frequencies, like nothing else. So they found that there are certain signatures that are very specific towards particular forms of cancer, right? And they did this for, I think, 33 cancer types. Like up to now, it's a 2013 paper. So right now they have published many papers, and they have covered all the major cancer types. And so this was our primary motivation, and this because we thought that maybe we can utilize a larger sequence pattern upstream and downstream. Instead of just taking one, we would take an arbitrary number of nucleotides upstream and downstream. So maybe 5, 6, or 10, and maybe try to look at whether this would be able to capture the information in the mutation. So can we use this particular sequence in the upstream and downstream and predict whether this is a driver or a passenger mutation? So that was our primary motivation. Again, this is also a very relevant paper that I should talk about. So this is a PLOS genetics paper. Again, it's a bit old, but it has been a very essential paper for us in terms of finding context of mutations in cancer genome. So what this paper did was they looked at melanoma patients, right? So they looked at melanoma patients, and they concentrated on the promoter regions of the genes that are implicated in melanoma, right? So what they found was there is this recurrence pattern of TTCCG whenever there is a cytosine getting mutated. So they looked at the promoter region, they looked at all the cytosines in those promoter regions, and what they found, this is a specific position in that promoter region where a cytosine had a tendency to mutate to something else. And they found that whenever there is a cytosine getting mutated, they would find this pattern, TTCCG, right? And this recurred across patients, but only exception was this third gene, so TERT third gene, where this pattern was not followed. So again, there were a lot of follow-up papers that explored this for the reason why this was the case. And so this was also essential because what we were looking for is do the same thing for driver and passenger mutations. Can we look at these mutations and sort of say that, okay, this is the particular context that would be specific towards drivers, and this is the particular context that would be specific towards passengers, because that would give us an entirely new perspective on defining these two terms, drivers and passengers. Because up to now, there were statistical models that were coming up, but very few papers have actually concentrated on utilizing the context. And also this was one of our motivation to go for this work, and we formed a hypothesis based on this. So our first hypothesis was, if you look at this particular mutation, G, G, C, and C, so these are marked in red, can we look at an arbitrary number of nucleotides upstream and downstream from this position, build computational models, and sort of predict what type of mutation this is, whether G is a driver mutation, whether C is a passenger mutation, and so on. So the first thing that we need to show was there exists a difference in the distribution between the driver and passenger neighborhoods. So this is what we are calling as neighborhoods, right? The shaded part. So these are the neighborhood of this particular mutation. So G has this neighborhood ATCG and GTAC and so on. So we needed to show that there is a difference between the representations, the way we represent these neighborhoods, because these are all nucleotide patterns, right? We need to transform them into some sort of numerical format for our algorithms to work. So the first hypothesis was that there exists a difference between these two neighborhoods. The second hypothesis was, can we utilize this difference? So say we find any difference in this first part, can we utilize this difference to build robust machine learning models such that we can distinguish between these two types of mutations? So so say these are the two classes, so these are the two probability distributions of the driver and passenger neighborhoods, and we find that these are very different based on a particular p-value cutoff. We say that, okay, this is very different, and so we might have some signal there, and this might inspire us to further build machine learning models so that we can distinguish between these two classes. So first we needed to look for a signal, because that would mean that we should get on with this work. Otherwise, there was no meaning to sort of undertake this entire thing, because if there is no signal, if there was no signal in this within the neighborhoods, then we cannot build any machine learning model, because a machine learning model would utilize the differences in the features that you extract from the driver and passenger neighborhoods. So this was our hypothesis, and this was a workflow. Okay, so there are 3 different parts to this workflow, and okay, there is a lot of parts, but I would explain each to the best of my ability, because this is very straightforward. So you start with a bunch of mutations from some cancer mutation database, right? And then you extract these mutations and extract the neighborhoods. So after you extract the neighborhoods, you have to map them to some numerical format. So this is the pre-processing part. That's it. You have a bunch of cancer mutation. You need to extract the neighborhoods, represent the neighborhoods to in the form of a numerical. You need to find the best numerical representation that is possible, right? So this was the pre-processing part. The second part was to show that there is a difference, right, between the two feature representations. To do that, we use this technique called kernel density estimation. So this is part of machine learning models called generalized models. So this looks at the underlying, this tries to model the underlying probability distribution of the data, and instead of trying to fit lines and curves, which again falls into the purview of discriminative models. So kernel density estimation, what it shows is that these are the probability distribution, and we needed to show that there is a difference. This was followed by validation of the machine learning part, where you need to show that your model is performing well. So given we find a signal here, we needed to show that, okay, so we train our data on this, and we tested our data on something else, and we show that our model is generalizable. So it works well with all sorts of databases. By all sorts of data sets, by data sets, I mean mutations, right? Because that's our primary data. So we have a bunch of mutations, and we have the neighborhoods. So this is the internal and the external validation. We split into 2 so that we have the best criteria. We meet the best criteria that's possible. And there is no overfitting, or our model does not learn the patterns of the training data. And so these are the things that you need to take care when you're working with machine learning model. So this was the overall workflow. Coming to the data part, like what data did we use? So we had a data set from this particular study, Brown et al. So why this data set? It's because this data set was the only data set, to the best of my knowledge, which had mutations from experimental studies. So they have experimental evidence that this is a driver, this is a passenger, right? We need that experimental evidence, because if our mutations are from computational predictions, then it does not serve our purpose, right? So we are building ourselves building a computational algorithm. If we take our initial data set from some other computational algorithm, that entirely defeats our purpose. So our model would not be generalizable. So our model would not translate well to a new set of mutations that might come later in the future. So we needed to capture as much information possible from this experimentally validated set of mutations from 58 cancer genes. And we had a data set of around 5,265 mutations, among which 1,000 are drivers and around 4,000 are passengers. So these are the intermediary steps. So I'm sure you are aware of these different genome builds. So we extracted like sequences, like the raw nucleotide sequences surrounding a particular mutation from the GRCH31 build of the human genome, and we use this particular tools to extract those sequences. So this was the pre-processing, the feature extraction part. So that's it. We don't need any complicated way to represent our features. We just need the raw nucleotide sequence. Now the next part would concentrate on mapping these features or raw nucleotide sequences to a numerical format such that your algorithm can process it. Because up till now, these are just A, T, G, and C's, like series and sequences of A, T, G, and C, long sequences. These are not represented in a numerical format. So this was the next step. Uh, the first part was to look at the original sequence, which would be, say, this is a particular mutation that we would like to predict whether it's a passenger or a driver, and these are the nucleotides surrounding that particular mutation. So if there is an A, I would represent it as 1000. If there is a T, I would represent it as 0100. This is popularly known as one-hot encoding. So the particular nucleotide that's present, I will represent it as a 1 or else a 0. And I will have overlapping KMR representation as well, which would mean that AA, AT, TC, and CG, the overlapping KMRs of size 2, overlapping KMR of size 3, AAT, ATC, and so on, and similarly for 4. So why this representation? We wanted to check whether these overlapping KMRs would translate into some region of the protein coding genome, like such that we can actually pinpoint that these are the particular regions that might be of interest. Because like if you look at this particular one-hot encoding, biological sequences tend to occur in 3, right? So KMers get translated into codons, like codons get translated into proteins and so on. So like amino acids, and then they're encoded into proteins. So we wanted to have a representation that would capture this information to the best extent as possible. And although you might argue that AAAT is not very relevant, because there are biological sequences. I don't know whether there are biological sequences that are represented as dinucleotides. Although, yeah, CpG dinucleotide is also a well-known dinucleotide that's very specific to cancer patients. Like the CpG islands is also of specific interest. But we wanted to check how varying the number of KMers would affect our final model. So these are the different feature representations that we like to work on. So having got this representation, we have to map it to some numerical format, right? So this is what we did. So these are the two different techniques that we use. So one is the count vectorizer, and the second one is the TF-ID vectorizer. So these are very straightforward way to represent data. So for instance, we have this particular two set of documents. Consider that this is a document. So 1 gig helps 2 gigs and 2 gigs help 4 gigs. So count vectorizer representation would be this. So 1 would be how many times 1 occurs in the first document, how many times 2 occurs in the first document, second document, and so on. So this is just counts the number of times the token. So these words are individual tokens. Of course, in our case, it was groups of nucleotides, 2, 3, or 4. So this was a word matrix. And again, TF-ID vectorizer is slightly a modified version of count vectorizer, where they use the tokens, but they also introduce an IDF term, which IDF term captures rarer words. So there is this particular term in the denominator, right? So frequency, more frequent words across that are occurring across the documents are given less priority. So and the them, etc. These are all words that are not very interesting, right? So if there are certain words that are very rare, that is specific to a particular document, then those words would be captured by this inverse document frequency. So we would derive this scores in the form of count vectorizer scores and TF-IDF scores. So this particular representation would be transformed into this particular matrix. So this would be our final word vocabulary, KMR vocabulary. Like mutational neighborhoods would be the rows, and these are the KMRs that are possible, right? So 256 KMRs that are possible. So right. of 64 kmers for kmer size of 3. If you have 4, then 256. So this would be 256 columns and 5,000 rows, which would be our initial data set. So this was the final data metrics that we would feed to our machine learning algorithms to build the final models. So I would quickly go over this kernel density estimation. Like this is, I've already talked about how this can be used to sort of pinpoint differences and similarities between two distributions. Well, not differences. Kernel density estimation is specifically aimed at trying to jot down similarities between two distributions. So if two distributions are very similar, they would get a score and a lower score, and if they're very different, then they would get a higher score. So or okay, so vice versa. And so this was the reverse of what I said. So if you have a very similar distribution, you would get a lower score and so on. So why this particular technique? Because we wanted to check whether the driver and the passenger neighborhoods, so these are the contour plots of the distributions, how much different they are. So if we could calculate or quantify this difference somehow, then that would be the validation of our signal that we were looking for since the beginning, right? So that neighborhoods of drivers and passengers are really different. So the next thing that we did was we built machine learning models and we tested our algorithm on this 989 literature curated mutations. Again, by literature curated, we did not want any computational predictions. We wanted mutations that had literature evidence of some sort across 15 cancer genes. And we tested how our algorithm is performing as in comparison to this 11 pan-cancer based mutation effect prediction algorithms. So that would give us a clear indication of how our model is performing. And this was the number of drivers and passengers that we had. And the final model that we would get after filtering out this number of mutations, because we cannot have these mutations in our training data if we are testing it on this mutation set of mutations. So we filtered out this 989 mutations. And the final model that we built was, we named it as NBDriver or neighborhood driver. And we tested it on this different metrics. So how sensitive it was, how specific it was, what was the positive predictive value, and what was the negative predictive value. So these are all the different metrics that I talked about in the beginning that are essential for validating or comparing our machine learning algorithms with the other models. So this is what the first results that we got. So we found that the difference between the driver and the passenger neighborhoods is significant. So all the stars that you see, except the neighborhood size of 1. So these are the different neighborhood size, right? So 2 means 2 nucleotides to the right, 2 to the left. 3 means 3 to the right, 2 to the left. So again, there is no right and left in the genome, but since it's a 2D projection, as I'm presenting, so it's an upstream and downstream for the mutated position. So once you get this, we found that okay, so there is some signal in our data, and this sort of motivated us to further pursue our machine learning part. So we saw that there is a difference, except for one window size, there is a significant difference in the distributions between the drivers and the passengers across different window sizes. So the highest we tried was 10, 10 to the 10 upstream and 10 downstream for the mutated position, and the next part would talk about the comparison. So we built machine learning models, and what we found was our model NBDriver integrated. So this is integrated means we added features, our neighborhood features as well as other features from the other most commonly used mutation effect predictors. And what we saw was our model was performing reasonably well, provided that it's using a very simplified representation of features. It's not using any complicated representation. It's just using the features from the raw nucleotide sequence, and it's trying to build models. And we achieved this results using just 50 features, which, if you're familiar with machine learning, so the lesser number of features would imply lesser model complexity, at least to some extent. So we had just 50 features, and using just these 50 features, 26 out of which were neighborhood features or the raw nucleotide sequences, we could accurately predict drivers from passengers with this much accuracy. And we also, one important metric that I would also like to point out is the negative predictive value, which is very essential to ward out mutations, passenger mutations. Like negative predictive value tells you that what are the like if your model is predicting a particular mutation as passenger, what is the likelihood that it's actually a passenger? So that is also important, right? Not only we should not concentrate on the positive class or drivers in our instance, we should also like devote a sufficient amount of time modeling our algorithm so that it takes into account the negative class as well, right? So we also had a decent negative predictive value if you compare it across the different prediction models that you see on the screen. So as far as our ability to predict unseen pathogenic coding point mutations, our model performed reasonably well again in terms of all these 4 different mutation data sets. So this is the cancer mutation census. It's a very well-known data set from Cosmic, the catalog of somatic mutations in cancer. Canra is another tool that a similar tool like NBDriver, and they have published this list of mutations which were rare variants, rare mutations. Our model could perform well in terms of only one cancer type. So GBM is glioblastoma multiforme, so it's a aggressive form of cancer that affects the brain and the spine, and OVC is ovarian cancer. So in terms of NBDriver could accurately predict 85% of unseen rare glioblastoma multiforme mutations and 53% of ovarian cancer mutations. It's not performing well in ovarian cancer, and that's sort of leaves room for further analysis. And NBDriver ensemble is another technique that we use. We combine the predictions of NBDriver and other mutation algorithms, and we found that that significantly improved the accuracy in terms of this rare driver mutation prediction. So why it's important? If you remember the first slide that I showed, rare driver mutations are the ones that are singletons, which occurred only once across all the patients. So that's why it's important to take into account to actually check whether your model is performing well in terms of the rare variants as well. So one thing that we identified is based on the mutations that we correctly predicted, we identified 138 driver genes. So the mutation, a driver gene should at least have one driver mutation, and we identified 130 driver genes, and 130 out of 138 driver genes were already they had an overlap with already existing large-scale driver discovery studies. So they were already well-known cancer driver genes. So that's a nice indication that the mutations belong to genes that already had literature evidence from multiple sources. And the 8 remaining genes had functional roles in tumor development from several studies. So these are the 8 genes that were not present in this large databases or driver discovery studies, but had functional roles in progression and development of tumor. So this is the validation part, like how well your model is performing, and these are just accuracies. I've just these are whatever I see on the screen are how much the number of mutations correctly predicted divided by the total number of mutations. Just the plain good old accuracy of our model. And if you want to go and look into the more complicated scores that takes into account how your model is performing specific to machine learning. So these are all the scores that we use: sensitivity, specificity, and so on. Summarize. Okay, so you have a bunch of mutations, and so this is more or less the workflow of our model. So you start with a bunch of missense mutations. You try to extract the neighborhoods upstream and downstream from the mutated position. You try to find clever ways of representing the features, the neighborhood features. Then you try to show that these two are different, like the driver and the passenger neighborhoods are different. Then, inspired by these, you build machine learning models, and you develop this tool NBDriver, which is all perform better, which performs reasonably well in terms of accuracy, how accurate it is at finding pathogenic alterations across multiple driver discovery studies. So this was our major findings. We not only found signal, we did build models on top of the mutations that we had, and we found that our model could accurately predict missense mutations that were unknown, like were not used to train our model, like unseen coding point mutations. And yeah, so this was more or less what our model did. And in terms of key results and contributions, I've already gone over all of them. So except one window size is fine. We found that there is a significant difference in estimates. These are the results. We have got an accuracy of okay, so we got an AUC, which is again a machine learning metric, so of 832, and MCC, which is again used to calculate how in cases where there is an imbalance, this is a metric that is used. And we also got reasonable performance in external validation. We got an accuracy of 0.89, and we found that 130 out of 130 driver genes had an overlap with the existing studies. So this was very recently published in the journal Cancers. So I would encourage you to go and have a look, which details out each of the steps that were taken and all the problems that we faced while building the initial training data, the problems that we faced while uh, jotting down all the different validation data sets that might be of help to validate our model. So there were a lot of problems because this kind of studies require mutations from databases that had experimental evidence. We did not just want to build our models on someone else's predictions, because that would mean that our model would not generalize well. So, but we did find a data set that had, I won't say it's a huge data set because it's just 5,000 mutations, and by number of mutation standard, it's very less. If you look at Cosmic, they have millions of mutations, cancer mutations, but this 5,000 mutations captured the maximum information possible, which is again exhibited by our superior performance in the external validation experiments, where our model has not seen any of the coding point mutations and did perform reasonably well. And this also got reasonable media attention because this was again very recent, and there was this IITM TED talk that recently came out last week. Again, this was a popular science piece on whatever we did, and I would encourage you to go and take a look at it. Now I would move to the final part of my talk, which is precision cancer genomics. I would devote 1 or 2 minutes to talk about this. So this is basically what I hope that many of you would be interested in the future to work on. So you have this different patient-specific data, like mutation data, mRNA data, and so on. So to extract features from these different modalities. So these are the different data modalities. You extract these different features, and you try to build machine learning models like we build. These are the already existing models, right? These are patient-specific driver genes. So this is again an active area of research where you just look at a single patient and try to pinpoint what are the driver genes in that particular patient instead of looking at a cohort. So you can run these features through this particular algorithms, which are machine learning models. Based on the output, you can query large databases and generate comprehensive reports, which you can again choose for clinical decision making. So this is the entire idea of precision cancer genomics. So how does it translate into practice? So the clinical aspect of this, the translational capability of a model is also very important, and that is only possible if you have built a model and have done the additional step of actually validating it across multiple cohorts, such that any clinician or practicing pathologist who might be interested in looking at this group of mutations or your predictions specifically might feel interested and actually motivated to go and have a look, because you have a model that is generalizable, that actually translates across multiple data sets and not specific to a particular data set. So I keep like stressing this point because there are several models that come out that just do some sort of cross validation inside their own training data. Like they just take their own training data, divide it into several parts, and keep one part aside for testing and report the results. But that is not the right way. At least if you want to see your model like translate the translational aspect, if you want to concentrate on, this is the right way to do. You should query large databases. All this like Cosmic, Civic, OncoKB, these are the databases that has experimentally validated mutations from different cancer types, and actually see whether your predictions make sense, right? And you can also develop all sorts of web servers and so on, so to generate comprehensive medical reports. And finally, these are the 3 major contributions at the work that's going on in our lab. So the first one is iComic, which is a generalizable pipeline to work on cancer, like to pinpoint cancer variants from large omics data set. So if you have a large sequencing file, you just have to supply the sequencing file to a very well user-friendly sort of way. So it's a GUI. So you don't have to worry about the inside details, like what's going on in the inside. Also, like I would stress that you need to understand the different parameters that are going on, like that are required to run each of these tools. But it's very user friendly. You just have to. It's a drag and drop interface. You supply your tool, like supply your data, and it would give a set of predictions, and it would also run NBDriver in the background. And this other tool called CTAC, which is again a multi-omics tool, which the sole purpose of this tool is to build machine learning models to differentiate between the different types of driver genes, cancer genes. So the TSGs and OGs are tumor suppressant genes, and oncogenes are the 2 broad classes of cancer genes. And this is again a machine learning model that deals with genes and not mutations, like in my case. So these are 2 different data layers that we have concentrated on. Like these are the 2 major work that's going on, and also we have not come up with a fancy name for this work. So it's identifying co-occurring gene mutations in cancer. Again, this is a multi-omics work. So it looks at this complicated network representation of different co-occurring gene mutations across cancer, multiple cancer types. It's a pan-cancer model again, and it tries to find out whether there are functionally relevant pathways. There are certain biological pathways that get affected if 2 mutations are switched on at once. So these are the three major projects that are going on in our lab, and I would encourage you to go and take a look at it. All of this, at least the abstract of each of these work is online. And finally, I would like to acknowledge my supervisors, Dr. Karthik Raman and Dr. Ravindan, for their help and support throughout. And I'm also part of these two centers, Center for Integrative Biology and Systems Medicine and RBC and I would also like to thank my lab members. I'm part of the Computational Systems Biology Lab with Dr. Karthik Raman as the PI, and uh, I really valued from the suggestions to our regular group meetings and so on. Many meetings just like this. So yeah, thank you. I don't know if I've missed the time, but yeah, I would be happy to take questions.